session will be focused on understanding the dynamics of marine species and how that's relevant for management. I think we heard from all of our presentations from the states that it's really one of the first questions that come up in terms of how to address and deal with the entanglement issue is we need to know more about where the whales are and what are they doing. So that's a big reason why we're not only starting with this in terms of a scientific the presentations on the science, but while we'll have actually 2 sessions looking at this issue. There certainly is a lot of research, even prior to the entanglement issue, on understanding whales on the West Coast, and so there is quite a bit to draw from. But I think we don't know, everything all the time, or we certainly don't know as much as I think we'd like to know. Particularly, it seems like when we want to know it, we seem to be short on a bit of understanding. So anyway, really looking forward to this session and learning more about the most recent developments and what we know about whales and sea turtles here on the West Coast and how that's relevant to management. Just quickly before we get started to kind of review the process here, what's going to happen is we'll have a series. In this case, there'll be 5 presentations, all of the 10-minute variety. Then we'll take a break. And after that, we'll come back and convene a panel that will include the presenters as well as a handful of other folks for a group panel discussion. If we have time, we'll pause for some clarifying questions. As we move through the talks, given that they're pretty short talks and our time frame, it's highly likely we'll not be able to incorporate too many Q&As, but we'll do what we can here. So in this session, you're going to hear 5 excellent talks from some really smart people here, and they're going to be focused on the, what we know about the, the general ecology of whales and sea turtles here on the West Coast, and in particularly looking at some of the monitoring programs and ideas for monitoring these things moving forward. We're going to have John Kalambakidis from Cascade Research presentation. Karen Lohman from Oregon State University as well, Daniel Palacios from Oregon State, Leah Torres from Oregon State, and Scott Benson from our NIMS Southwest Fisheries Science Center. As we were doing in the other session, participants, you'll be able to submit your questions through the Q&A function, and we'll do our best to incorporate those, if not during the presentations, but as we get into the panel session, as we do. For your presenters, as you've been fair warned, we're on a tight timetable here, so we're doing our best to hold you to that. You will have those cues that you'll be able to follow in terms of when you're 2 minutes left in your talk and when time is up, and so we'll certainly check in with you if we get to that time's up spot. So without further ado, let's start with John Kalambakidis from Cascadia. Take it away, John.
Great. Thank you very much. I want to first just acknowledge a number of people. Some of the information I'll be presenting here has been in partnership with a number of groups and entities. I've listed here some of the researchers with Cascadia and SR3, but there are many other collaborators that are part of this. And much of the work I'll be reporting on has been supported by different divisions of NOAA, and in particular recently through a Section 6 NOAA funding that goes through the Washington Department of Fish and Wildlife, but help support some of the work up and down the U.S. West Coast for us. Part of what I'll be trying to do is touch on a little bit of background on, in particular, humpback whales that might help introduce some of the other talks and be relevant, especially the genetics talk, but talk a little bit about the role of population monitoring in insights into the entanglement issue. And as a part of that, I'll be updating some of the population trend data we've been generating, some of the units of populations that are being monitored, how we're using humpback whale entanglement scars on humpback whales as a tool. Some of the planned research we have to better examine some of these questions relevant to the Dungeness crab fishery. So I'll be trying to touch on those as we go through this. One of the long-term elements that we've had going since the 1980s has been a long-term photo ID program on, in particular, humpback and blue whales, but also including gray whales, and particularly the Pacific Coast feeding aggregation of gray whales. Those efforts have identified thousands of unique individuals, over 5,000 in the case of humpback whales, and documenting some 50,000 encounters along the U.S. West Coast, using these unique markings on whales, and then using these to look at movements and abundance that I'll talk about. Most of our survey effort has been done from small boats, such as you see here. This is from 2018. You see some of the small boat tracks, some of the sightings of humpback whales, and biopsies where we collected samples just in that 1 year up and down the coast. And you see we cover pretty much all of the West Coast, operating out of different areas. And humpback whales are distributed all along the West Coast as well. Some of that information in the past has been used to inform some of these biologically important areas. I'll emphasize that there are some areas of concentration. Humpback whales are concentrated particularly along the continental shelf, up and down the coast. We do divide Washington and southern BC into a separate feeding unit, but they occur widely. All those dots you see on the map are actually sighting locations that we've worked with it. The density of whales off Oregon and Washington is a little bit lower off than some of the areas off California, but they do have very large populations, and I'll show you some of those numbers. A key part of the photo ID work is how it feeds different aspects of the entanglement question, and in particular, the abundance and trend data that we get from the mark recapture estimates that I'll focus on for a second have served as the basis for the estimates used by NOAA that then calculate the PBR or the potential biological removals that trigger some of the concerns about fishery-related impacts. So that case plays a key role in that. Our migration and movement data, and especially how these whales move to different breeding areas, is key in how it intersects, especially with the Endangered Species Act and some of the different distinct population segments that have different statuses under the Endangered Species Act and are based on what breeding area whales go to. And then we also use these photo IDs to track the different individuals that get entangled, something you'll hear more specifically about from especially a presentation by Jen Tackaberry later on in a later session. But these are what allow us to look at things like survival. And I'll cover a couple of other aspects as well, but these photo ID parts, this is what they fit into. Just to look at the context, abundance of humpback whales, this is our latest abundance estimates, 3 different models for humpback whales off of California and Oregon, showing the pretty dramatic increase that we've had. Overall, and hopefully you can see my cursor, but the blue dots with dashed lines show a 7.5% annual rate of increase, which is about the trajectory these whales have been on, though we were initially puzzled by what seemed to be a stabilization and leveling off of the population in the early 2010s that now has been followed by this rapid increase. We actually think it's more likely that there wasn't a lull and the population was increasing at that rate throughout, and that appearance of a leveling off and then a rapid increase the truth is probably in between those two, and they've just been kept increasing. But that's a very key factor in understanding, at least one part of the equation of why entanglements are increasing. Along with this, we've certainly seen expansions of humpback whales into areas. This is just to show you we have separate estimates for Washington and British Columbia. They similarly are showing an increase in trends between the two now, over 5,000 humpback whales along the U.S. West Coast. We also generate similar abundance estimates for some of the other species like blue whales that you see in the lower graph that are a little bit in contrast to humpback whales, but have shown a bit more stability in terms of population trends, but also a little bit of an upward trend in recent years as well. Now, in some specific areas, like this is data updated for the Salish Sea in Washington, and you'll see a much more dramatic rate of increase in certain areas. And this can be the case where humpback whales are potentially returning to areas they used to be abundant pre-whaling, but were wiped out and largely have not returned until more recent years. We've sometimes seen these more dramatic increases. Salish Sea coming into San Francisco Bay, expanding range in Southern California. There are areas that has further expanded humpback whales into habitats where they weren't common at a more rapid rate than the population trend indicated. Now, I mentioned these separate DPS units, and they are defined by breeding areas, partly because of the way the Endangered Species Act is listed. And so you have a Western, a Hawaii-based DPS, a Mexico, and a Central America. Central America treated as endangered. Those that breed in Mexico is threatened. Those that breed as Hawaii is delisted. But those animals go to different areas in different proportions based on their feeding areas. And there isn't a one-to-one correspondence between feeding and breeding areas. And in particular, we've documented that the proportion of animals going to these breeding areas, which is shown by these different bar graphs that correspond to the map, does vary as you move up and down the coast quite a bit. Highest proportions of endangered Central America whales off Southern California and decreasing as you come north. Some of the key research we've been trying to address involved looking at kind of this whale habitat distribution, migratory timing, but also, looking at some of the ways it links to the question of entanglement and rate of entanglements. One key element we're concerned about and are trying to help document is the degree to which entanglements that we currently document with just reports are severely underrepresented in the reports, and that degree of underrepresentation varies by region. Okay, I'm going to go a little bit quick here just to say we have a research program supported by the Ocean Protection Council that will start this fall that will try to use some of these metrics to a greater degree. We'll be looking at not just the habitat whales use, but several ways in which we can examine what they're feeding on, including deployments of tags in some of those studies. The SCAR, we have two different SCAR studies going on that are informing different elements of it. The first one completed, but now a publication being worked on that was done as part of a master's thesis by Annabelle Wall, has actually documented the scarring rates from entanglements on humpback whales by geographic region. And the main take home in this table you see in the middle is that while there's some small variations in the average proportion of animals that have entanglement scars, either in any part using different types of photographs or particularly using the tailstock photos, those differences aren't very large, and they were not statistically significant in the models that Annabelle has developed. So it actually revealed that humpback whales up and down the coast face a similar risk of entanglement, at least based on these scars on animals that survive the entanglement. Keep in mind these are survivors of entanglement. We have a second study being initiated that'll be done by a student, Allison Payne, that will be looking at more the rate of scar acquisition over time and get at that rate. I'll point out that part of the goal of these scars is to use them to get a better sense of the true numbers of entanglements occurring. One of our big concerns is that by using only reported entanglements, those entanglement reports are very influenced by the reporting rate. So lower entanglements in Oregon and Washington, we know, are heavily influenced by the much lower reporting rate that occurs in those areas. Fewer whale watch activities, fewer recreational boaters, more wide open ocean, rougher waters out there. And that's really affected things as compared to an area like Monterey Bay, where there's an active whale watch and very educated and informed and high levels of reporting that occur. And it's important to remember our goal should be to decrease overall entanglements, but we actually want to improve reporting of entanglements because that's what gives us the information on what's really going on with the whales and how management efforts might be working or not. So my concluding or final points are just that, they're key metrics that we think measure the true number of entanglements that we're working on actively to kind of document that. And we think some of this population monitoring is a key way to get that, especially now that there may be disincentives to reporting on the case of fishermen, for example, if they're worried about the impact on closures. Population levels are increasing of humpback whales, and they're an important factor in understanding entanglement rates, and especially how the animals are distributed in habitats that overlap with the fishery. And then that need to increase entanglement reporting and the integration of that monitoring and documentation to allow better response. So I think I better stop there. And I think I'm close to time anyway, hopefully.
Yeah, thank you, John. No, that was excellent, and yeah, appreciate the job there. So, but we will not have time for questions in this moment, but we're going to pick up, I'm sure, on a number of those threads in the discussion later today. But thank you very much. All right, next will be Karen Lohman from Oregon State University. Karen, take it away.
Waiting for my slides.
I do hear you.
Yeah, Dana was that my slide.
Oh, here it is. Okay.
Okay, thank you. Hi, my name is Karen Lohman. I'm a master's student with Dr. Scott Baker at Oregon State University. We're part of the Marine Mammal Institute at Hatfield Marine Science Center. And I'm going to be sharing some of my work on the genetic population assignment of humpback whales in the East North Pacific. Next slide, please. Yeah. So as we just heard, management of mixed stocks is difficult. John Calamakitis was just talking about the GPSs and the genetically differentiated breeding grounds in the North Pacific. These have their own separate status listing under the ESA. Each of these populations or GPSs have their own abundance estimates, which means they also have their own potential biological removal calculations. And this is what makes management of the feeding stocks particularly difficult, given that we're still working to understand the degree and extent of GPS mixing along the US West Coast. The PBR calculations for that feeding stock may exceed the allowed PBR for one or more of the GPSs that compose the mixed feeding stock. And in light of the entanglement problem, this is really the crux of the GPS issue and why there's so much interest in understanding GPS assignment for humpback whales feeding along the US West Coast. Next slide, please. Genetic approaches are a really excellent way to assess this population connectivity because by employing population genetic methods, we're not limited to direct recaptures or monitoring the behavior of individuals. By using samples representative of the feeding grounds and from each of the breeding grounds, we can make inference about population connectivity through comparisons of genetic similarity between those sample sets and make inference about the migratory connection between the regions. The work I'm presenting today builds on the benchmark SPLASH study that was conducted between 2004 and 2006, which produced a snapshot estimates of genetic diversity across the North Pacific Oceanic Basin. The breeding ground samples collected in these efforts, which were used to help delineate the GPSs, are the references I'm comparing the feeding ground samples against. Next slide, please. Those feeding ground samples are composed of 765 tissue samples collected between 2002 and 2019. And as you can see, in addition to the extended time frame, the feeding ground samples represent quite a good geographic distribution. Next slide. A standard DNA profile was created for each of these samples, which produced directly comparable data between the feeding and breeding regions, which is sufficient for individual identification and population assignment testing. Next slide. So how the population assignment procedure works is that the DNA profile of each individual is compared to the representative breeding ground reference sample. And based on the allele frequencies of those reference populations and the unique DNA profile of the unknown individual, a likelihood of assignment to GPS is produced. So for example, using colorful whale number 1, This individual is largely blue, which indicates that the majority of its DNA profile matches to alleles that are more frequently found in the Central America reference population. This is an example of an individual with a high assignment to a DPS. Unknown 2, on the other hand, is representing an individual with mixed or weak assignment to DPSs. As you can see, there's no clear pattern that indicates matching to one DPS over another. And we find both of these instances in our data. Next slide. So if you didn't quite follow that, you could also think of this as 23andMe for humpback whales, or just like for humans, and just like you've probably heard in the commercials, we have an estimated percentage or proportion of genetic identity from a population for each of the individuals tested. The percentage or proportion of assignment data is the metric that we work with in further analysis. Next slide. And what this gives us is a way of assigning individual humpback whales to their respective DPSs. This is potentially quite useful in forensic applications of the assignment of injury or mortality to DPS, which would help us keep a more accurate tally for each of those PVR calculations. Of course, if it's being used in these applications, we need to have accuracy estimates. And this plot is showing the results of self-assignment testing using the breeding ground references. And you can see visually by the color blocking, as well as in the table, that we have pretty good accuracy estimates for Central America, Hawaii, and the Western North Pacific. Notably, Central America, which is the DPS of greatest conservation concern in our management scenarios, has the highest assignment accuracy. Assignment accuracy to Mexico is lower, and we think this may be due to unresolved breeding ground structure within that DPS that limits our genetic resolution. Next slide. So that's the individual level, but what about the population level? When we look at the distribution of feeding ground assignments in space, we're finding patterns of mixing that are consistent with other lines of evidence, indicating a higher probability of assignment to Central America at the more southern latitudes, higher probability of assignment to Hawaii at high latitudes, and assignment to Mexico that is distributed across a broader range of latitudes along the US West Coast. What this spatial analysis is intended to do is to help us assess the probability of a humpback whale being from a certain DPS in relation to time and space over the course of the feeding season. Next slide. And we've been working on this using generalized additive models, or GAMs. Using assignment probability as the response, we've looked at the effect of location, sea surface temperature, time, year, and month. We found that collection location, particularly latitude, was highly significant for Central America, Hawaii, and Mexico. Using this model formula, we have a high deviance explained for the Central America and Hawaii models, with a slightly lower but still good deviance explained for Mexico. And remember that makes sense given both the broader range of feeding latitudes that individuals from that DPS are documented at, as well as the lower confidence and assignment probability to that DPS. And what this, this indicates that there is a pattern to the spatial distribution of the DPS feeding along the US West Coast. And the hope is that this data can be used to draw informative lines on management maps. Next slide. And with that, I just want to thank all of my wonderful collaborators and the people who collected my samples and funders. Thank you.
Okay, thank you very much, Karen. I think we'll continue moving on, and we'll turn to Daniel Palacios from Oregon State University. Daniel.
Great, can you hear me?
Yes, I can.
Excellent. Well, thank you for the introduction and also for the opportunity to present on our science from the whale tagging perspective and how it can inform entanglement management. I especially want to thank Jen Humberstone and Dan Lawson for the encouragement to participate. Let's just go back for a moment. Or okay, you can keep it there, it's fine. Before I dive into the presentation, I just wanted to provide my objectives for today, which are three. One is to provide a retrospective view of the substantial data sets that we already have that have excellent potential to inform management needs. Two, to provide a simple framework or vision for how we can achieve this by showing some examples of things we have already done, plus a sneak peek into some new and exciting methodological and technological developments. And three, lead us into thinking how the approaches that we have developed can be implemented into the future as monitoring tools that will be necessary to assess how management actions are working. So let's go with the first slide, please. And the idea here is very simple framework or vision for how we go from, we have a problem, in this case, entanglement, and we first of all need data. And then as we acquire more data over time, we're able to generate products that eventually will support decision support tools. That's the last box there that you see. And we see this commonly applied, for example, with abundance estimation, survey work, siting surveys, and so on. What I want to do today is show you how we can get there too via tagging data. So in our case, what we have been collecting is tagging data, and we have a secret weapon with tagging, is that from telemetry data, from tracking data, we can derive movement syndromes. Movement syndromes is a collective term to encompass a variety of behavioral movement behavior metrics that can be derived that are typically hidden in the tracking data. That is that until we apply some methodological tools, we cannot really see those patterns. But once we do, we start obtaining interesting information about the movements of the animals. In addition to that, we can similarly get to abundance and distribution using species distribution models, of which there are several off the West Coast for whale species. And if we're able to get there, we can finally get to risk assessment models by plugging these species distribution models or other parameters derived from the whale data into risk assessment. So let's just go one click forward. And all of these boxes, all of these phases are iterative processes. As we collect more data, we grow the information that then we can derive better products and better risk assessment methods. Next, please. One more click, there we go. So in the case of blue whales here, we have used tracking data that you see in the upper left panel for blue whales. In this case, we have 104 blue whales that we tracked between 1994 and 2008. And we have applied a variety of movement syndrome approaches to infer movement behavior, that is ARS behavior, which is indicative of foraging, or transiting behavior that is indicative of migration and movement between areas using state-space models, as well as home range analysis that you see there on the bottom, and other habitat modeling approaches. In the center, we see a couple of approaches that have been applied to this tracking data to derive abundance estimation for blue whales, in this case, from the tracking data and using oceanographic environmental variables. And then one more click, please. We have gotten all the way to risk assessment models. In this case, we have Abrams and Redfern as a couple of examples of implementation of products derived from tagging data to get all the way to make some apply some risk assessment methods, in this case for ship strikes in Southern California for blue whales. Next slide, please. So what we have here are 4 species: blue, fin, gray, and humpback whales. And this is data. You can see the years. This is what we call our historical data set that spans the years 1994 through 2008 or 9 in the case of gray whales. And this is thanks to the pioneering and visionary work of Dr. Bruce Mate, whose data I recently inherited. So at the scale of the West Coast, we have these data holdings, especially good for blue whales. For fin whales, in this case, we have a couple of examples. Gray whales is pretty decent, and you can see the coastal sort of linear behavior of their movement compared to, say, blue whales, which is all over the place. And then finally, on the far right, we have humpback whales, again, with some somewhat decent coverage. In the blue and purple blobs that you see there, those are areas of interest to conservation. In purple are the the BIA, the biologically important areas that John C. was talking about earlier, that he was leading the effort in development. And then in the blue are the national marine sanctuaries of the West Coast. Next slide, please. So since 2012, for the same 4 species, in support of annual monitoring requirements on behalf of the Navy, we have been collecting additional data. As you can tell, we have a comparable data set, new data that has not yet been published for blue whales, but substantial data for fin whales. You can see much more coverage up in Oregon and Washington, where we didn't have before, additional tracks for gray whales, and a lot of data for humpback whales of the West Coast. These data are largely analyzed, and we are looking for support to provide these, to conduct these efforts. So this presentation is as much a sales pitch to anyone who has an interest in supporting our vision to how we can take these data from where we are into some interesting applications for management. Next slide, please. That was at the scale of the West Coast, but even at very fine scales, we're looking at about 100 square kilometer area here off Coos Bay, Oregon, in southern Oregon. Our data, this is the same 4 species in the 4 panels, bluefin, gray, and humpback whales, and those same tracks. And even at this scale, you can see differences in the species. You can tell that we have somewhat decent coverage, especially for some. So we can come at the problem at various scales from the full West Coast to fairly small areas. Moving on to the next one, please. I mentioned applying tools for inferring movement behavior. So in this case, these are our humpback whale tracks showing colors for locations based on behavior using state-space models. So we have area restricted search in red and in blue, transiting between feeding areas. This is just for the feeding season of the West Coast. For 3 areas where we have tagged of California, of Oregon and Northern California, and then in Washington waters. And let's move on to the next one. The idea here on the map on the right, what you see is the aggregation of locations for our complete data set, and where that coloration indicates the density of locations. And then on the left, what you see is that at the scale of the different tagging areas, again, California, Northern California, and Oregon, and Washington, we are seeing that there's some differences in behavioral mode. Of course, it's a very coarse look at the data. Let's go to the next one. And what we can see is that we can slice and dice the data into finer boxes. In this case, we're taking 2-degree boxes along the coast, sort of sections or zonal areas like we saw earlier, but in this case, they're just arbitrary 2 degrees in relation to environmental conditions or other conditions, the physiographic conditions of the landscape, like in this case, distance to shore by latitude, or in the next one, please, sea surface temperature. And like that, we have a suite of environmental data that we can start looking at differences along of habitat use along the West Coast that can inform models. Next one, please. We can also, as I mentioned, look at home range analysis, and the overlap in home ranges for each tract individual can give us an idea of important areas of aggregation. So as you get into darker shades here, darker shades of brownish, sort of burnt orange color, that's where we have greater and greater overlap in whales in these areas. So we essentially are identifying important hotspots along the West Coast for animals that we track on average 3 to 5 months. Next one, please. Finally, this is I saved the best for last. This is the most exciting data set that we have. This is thanks to a technological development. Since 2016, we have been using a dive monitoring tag. So we can, over the same timescales of multiple months, we can get information for every dive in terms of depth of dive, number of lunges per dive, dive duration. And what you can see in the colors here in the top panel, I've chosen to represent the depth or dive. So this is really important for management because we are actually getting a spatially resolved vision of how the animals are using the water column at the scale of the habitat of the West Coast for 3 species for which we have been using this tag since 2016, as I mentioned before. The bottom row is just an indication of how many dives were monitored or recorded for those tags. So yes, I mentioned this is really useful because now we can not just look at the horizontal habitat use in terms of the movements, but also in the water column, in the vertical. We can look at diel variability in terms of time of day, as well as scales of days and weeks and months. And one more, can you please tell me where we are on time?
You got time for one more, Daniel?
Great. So this is an animation, and if you can just play it. I chose to show this animation because looking at maps of tracks doesn't really do the data justice. What we see here, colors are our tag deployments in California in red. In blue, you will see Oregon appear in a moment, and then in sort of light orange, tag deployments of Washington. So what you can see is the scale of movements of the West Coast for these animals and how they're ranging during the feeding season and then as they start the migration. Again, one of the interesting things about the tracking data is that we can see for individuals how they're using their habitat, whether they're coastal or offshore as they're migrating. And you will see in one case in a moment an animal returning from its migration back to the feeding areas. Sometimes they migrate along the coast on their way down to Mexico and Central America, and sometimes they come back offshore. And this case that we're looking at right now is actually an animal coming, migrating far offshore until it gets to the breeding area of Mexico. And then this is the animal which is coming back. Then it's taking a more offshore route. And I think that's the end of the animation. If we can please just move to the acknowledgements really quickly. Yeah, I'm not going to have time to cover this, but we can discuss this. But yeah, just to acknowledge that a lot of this work has been funded by the Navy, especially in the last several years, as well as a number of other agencies and private donors. Thank you very much.
Yeah, thank you very much, Daniel. Okie doke. Moving on. Leah Torres from Oregon State University.
Hi, yeah. If you can give me permission to share my screen.
I
There we
think
go.
it's been granted.
Yep, I just got it. All right. Great. Can you see my presentation?
I can.
Great. Okay. Great. Thanks. All right. Well, I'm really happy to be here today to describe this project that we have going on. I want to start by emphasizing that the origins of this project are really grassroots. They came out of discussions at our that the Oregon Whale Entanglement Working Group meetings a couple of years ago, just that highlighted the need for this research. So it's a really great partnership between my research lab, the GEM Lab at the Oregon State Marine Mammal Institute, the Oregon Department of Fish and Wildlife, the U.S. Coast Guard, and Oregon Sea Grant. And I really want to acknowledge our funders for this work, which has been from the Oregon Dungeness Crab Commission and NOAA Section 6 funding through the Oregon Department of Fish and Wildlife. There we go. Okay, so I'm going to start by describing our project steps that are leading to our objectives. So the first step is conducting these helicopter surveys for whales in Oregon coastal waters, where we record presence and absence data. Then once we have these data for 2 years of these surveys, we're going to relate that presence and absence data to a suite of environmental data to develop density distribution models, as well as to gain a better understanding of whale ecology in this region. And then from these models, we're going to develop seasonal whale density predictions, kind of like this made-up map that you can see here. Now, while we're doing these helicopter surveys, we also have a pretty extensive outreach program going to try and encourage citizen scientists or all ocean users, so whether that's the Coast Guard or tugboat captains, fishermen, or regular, recreational boat users, to use this mobile app called Whale Alert to record their opportunistic sightings of whales. And with those data, we plan to validate our predictions. And then once we've done that, we'll be able to hopefully revise our models and prediction maps and develop uncertainty maps. Now, at the same time that we're doing this, our partners at Oregon Department of Fish and Wildlife are using their logbook data to develop seasonal fishing effort density maps, like this map here that you see of Dungeness crab fishing effort. And so once we have both of these products, the whale density maps as well as the fishing effort maps, we will compare the two through an overlap index in order to generate seasonal predictions of spatial entanglement risk. And that's really what we want to get to provide managers with a tool that they can use to make regulatory decisions about where fishing effort should maybe and should not happen. So, but unfortunately, we're not at this stage of the project yet. We are only in this beginning phase. So the rest of my talk will really focus on this data collection side of things. So these are our 4 survey track lines. We work with the U.S. Coast Guard out of 3 bases, one in Warrenton up by Astoria, and then in Newport and in North Bend. So the black lines are the survey track lines. And in the left panel, it's the track lines are overlaid on bathymetry, and in the right panel, they're overlaid on a density map of a Dungeness crab fishing effort over 3 years. So our track lines have to be less than 150 nautical miles in length, and they have to return to the same port that we left from. In designing these track lines, we wanted to mimic sawtooth track line, and we also wanted to cover varied bathymetry habitat. So that's the left panel, as well as different levels of effort and fishing effort. So that's that right panel there. And our goal is to survey each one of these track lines once a month, so 4 track lines a month. Our protocols when we're flying are to fly at 90 knots, 500 feet. We have 1 observer that is also the data recorder. We use C-Scribe to record our survey data and sightings. We use distance sampling methods, and we're generally in passing mode while we're on survey, but occasionally we break to idea species if needed. These are our survey platforms. So when we fly with the North Bend sector, we use this Dolphin, which is slightly smaller helicopter than the Jayhawk that we use when we fly with the Columbia River sector. So that's me in the bottom left there. You can see it's a fairly cramped space, but we do have good observation visibility off the port side. And then this is Craig Hayslip in the Jayhawk and the Columbia River sector collecting data. So this short video here shows Craig photographing a fin whale down there with, I think, some Dall's porpoise swimming around him. And this is just to give you a sense of what it's like in our visual observation capacity. So I want to acknowledge here that this partnership with the U.S. Coast Guard has been really phenomenal, and we're really appreciative of their support. It's been a great opportunity to work with them. So into the data, to date, we've conducted 51 surveys, and we've done, actually, it's a few more now that we've gotten a couple more in August, but we've been fairly successful at conducting the 4 surveys per month that our goal is. Of course, we have to deal with weather issues, and the Coast Guard has some limitations and mechanical issues every now and then. And we also had a break between March and May due to COVID, but we're pretty much back up and running, which is great. So what I'm going to show you now is some of the data that we've collected during these surveys, but I want to emphasize that these are raw data, so they haven't been standardized by sighting effort or conditions or environmental conditions. So these are the locations of the whales that we've observed, the major groups: blue whales, fin whales, gray whales, humpbacks, and unidentified. Again, on the left, the sightings are overlaid on bathymetry, and on the right, they're overlaid on a previous map of crab fishing effort. And what you can see is that most of our sightings have been in the southern range of Oregon. Here up in Astoria, we have, the northern range, we have very few sightings except for a cluster of humpbacks at the head of the Astoria Canyon up here. But otherwise, we have pretty big clusters of humpback whales and blue whales. Gray whales have mainly been distributed in near shore areas. And we've also seen fin whales distributed across the whole range. This is the distribution of our sightings by month. And what you can see here is that we have definitely had an increase in sightings between June and November. Our sighting rates have gone up mainly due to humpback whales and some blue whales. We also have a peak in March of gray whales there due to the migration. This is the distribution of whale sightings by depth. And the take-home message here is that most of the whales that we have observed have been sighted in depth ranges between 100 and 200 meters, but we have seen whales out to about 1,200 meters depth. This is just for humpback, so when we zoom in just on humpback whales, and on the bottom left is their distribution by month relative to depth, and in the upper right is their distribution relative to distance from the coast. And what you can see here is that during the months of July and August, when we did have a lot of humpback whales in the area, they do tend to be distributed in shallower waters and closer to the coast. This again is just humpback whales. It's a density map that's weighted by group size of humpback whales that we've observed. And again, here the point is that the sightings of humpback whales have been distributed more towards the southern Oregon range and in coastal waters, especially, or more nearshore waters, especially between May and September. And just briefly, I want to mention this other part to the project where when we do see aggregations of whales or, blue whales or humpback whales, we mobilize in a boat to go out and collect photo ID and biopsy data from the whales. And the photos go off to Cascadia Research Collective to augment their long-term databases that John mentioned earlier. And the biopsy samples are going to the Cetacean Genomics Lab at Oregon State University to help with DPS assignment. So to wrap up, I just want to emphasize that even though this project is only really in its infancy, we've already been able to contribute some good information to ODFW to help with their regulatory decisions. For instance, we've been able to refine our understanding of the depth distribution of humpback whales, which has informed their proposed regulatory package. And additionally, we have really strong engagement with the fishing industry here in Oregon, the Dungeness crab fishing industry in particular. And that's a two-way street of exchange of information where we've been able to, when we see aggregations of whales from these helicopter surveys, we can provide that information to the fishery in hopes that they might be able to voluntarily move their gear out of areas where there's high numbers of whales. And in addition, many fishermen actually report sightings of aggregations of whales to us to help us with our data collection efforts. So that's a really great partnership that I'm really thrilled about. Also, our outreach efforts are through trying to get people to use this mobile app is really great because we're trying to educate the public about the whale entanglement issue as well as getting them engaged in the project themselves. So our next steps are to continue these surveys, begin full-on data analysis, and to produce the models and predictive maps to really help management decisions. So with that, I'll just put my email address up here to encourage people to contact me with any questions or comments or feedback. And again, I want to acknowledge all the great partners in the project and funding. So I think that's it. I'll wrap up there.
Excellent. Thank you so much. All right, moving on. Last but certainly not least in this section, we have Scott Benson, and he's going to tell us about leatherback sea turtles. Go ahead, Scott.
Thank you. Can you hear me?
I can indeed.
Wonderful. Pleased to be with you all today. I'll be speaking about leatherback turtles, particularly how we monitor leatherback turtles for entanglement risk. I'm Scott Benson. I'm with the NOAA Fisheries, Southwest Fisheries Science Center, and I've been working with leatherback turtles for about 20 years. Next, please. So we use a variety of techniques, a multidisciplinary approach to study leatherback turtles, genetics, telemetry, aerial surveys, and I'll be obviously focusing on telemetry and aerial surveys, and maybe some habitat sampling to inform key questions about movement, abundance trends, and foraging habitat. Next, please. So this is the big picture. These are telemetry tracks of leatherback turtles. They originate from beaches in the Western Pacific. They're part of the Western Pacific metapopulation. And in this image here, this is a state space model of movements. The darker colors there are areas where area restricted search occurred, which is a proxy for foraging behavior. And we could ignore for now the blue tracks, but you can see here the red ones are making a trans-Pacific movement from the nesting beaches over to North America. Next, just click one, please. And then in green are animals that were tagged in California. And so what you're seeing here is, in addition to this trans-Pacific movement, this what I call a yo-yo pattern between the foraging grounds off North America and then down to the Eastern Tropical Pacific or Eastern Equatorial Pacific and back to the foraging grounds. And that is because leatherback turtles do not reproduce every year. There's a remigration interval to the nesting beaches. It's about anywhere from 2 to 5 years. Next, please. So there's been a steep decline detected at the nesting beaches in Western Pacific. These beaches here that are got noted with the red arrow account for about 75% of all nesting activity in the Western Pacific. The most recent assessment by NOAA Fisheries in February 2020 indicates a 6.1% annual decline through 2017. Next, please. Likewise, we're seeing a similar trend here in California. In this case here, it's quite clear signal is in the inside of this figure here. You can see the Bayesian posterior distribution of the growth parameter lambda, which shows a high confidence in foraging population decline. The variability in here is linked to habitat variability, but also to nesting behavior and body condition. As I mentioned, these animals come back to these foraging grounds regularly before they go over to the nesting beach to reproduce. So one part of the population is over on the nesting beach, while another part is over here on the foraging ground. As a consequence of this decline, any bycatch is considered a concern, and NOAA has declared this to be a species in the spotlight as one of the 8 marine species most likely to become extinct, unfortunately, in the near future. Next figure, please. So here we're focusing in on the West Coast. And again, I'm showing telemetry data on the left panel. The red dots indicate areas of area restricted search or foraging habitat. So we've got 2 primary foraging areas, shelf waters off of Central California and shelf and slope waters off Oregon and Washington. These animals are present in these areas during as early as April through December, with the greatest abundance occurring during July through October. So that might be some good news for some of the crab fishery, at least. Critical habitat was established in 2012, and in the left panel there, the cross-hatched areas of Oregon, Washington, and Central California. And just as a reminder, leatherbacks do face risks from other fisheries. The red polygon there is what was known as the Pacific Leatherback Conservation Area that was established in 2001 to mitigate leatherback interactions with drift gillnet swordfish fisheries. Next, please. Thank you. So our data here from aerial surveys here, this is a long-term one on the left using actually harbor porpoise aerial transects going out to about 50 fathoms. You can see the greatest abundance occurring between Point Pinos and Point Arena. We also use what we call fine scale surveys to help us spot leatherbacks for capture and subsequent tagging. The distribution does vary a little bit from year to year. However, most of the turtles are encountered between Monterey Bay and Point Reyes, Bodega Bay area, with the distribution centered over the 50-meter isobath. These animals have been very well behaved in that regard. Next, please. At least in California. So the good news is we do have the ability to monitor these animals through tagging and aerial surveys. So as an example here, I've got on the left side, an aerial survey showing the harbor porpoise lines and the fine scale lines with the green stars being leatherbacks that we have seen. If we tag the animals, we can look at movement in real time, as indicated in that middle panel as well. And depending on the tag longevity and the attachment duration, when the animals do come back in the subsequent spring, we're getting more data about where these animals are occurring when they arrive at the foraging area. Next, please. Now, leatherback turtles are a very cryptic species. Unlike whales, they're very difficult to find, even from the air, and from a boat, it's almost impossible. So we use these biological indicators often to help us find these animals in neritic waters off Central California. Leatherbacks consume almost exclusively brown sea nettles in Central California waters, and these surface aggregations can be observed from aerial surveys. Likewise, we also use the mola mola, the ocean sunfish. It's a jelly predator, and they are also easily detected during aerial surveys, and this has been very helpful to us. Next, please. So this skews the busy slide here, but our effort off of the Pacific Northwest is much more limited. We just haven't had the funding support to be putting lots of resources into this area. But here's the data that we do have. It's clearly an important foraging area. The small green dots show the telemetry positions from 3 turtles that actually did trans-Pacific movements from the nesting beaches into these waters off the Pacific Northwest. We've had a few aerial surveys as well from our lab, shown in blue are the transect lines, and the black dots are the actual sightings of leatherback turtles. And we also have data from opportunistic sighting records, vessels, and aerial surveys, and then some bycatch records from the drift net fishery in the mid-late 1980s. So although the area is larger compared to Central California and the turtles seem more dispersed, monitoring could be based on the same approaches we've used in Central California. We're currently working on developing capacity, including finding sources of funding and local vessels to target this area in the near future. That's all I have that is pertinent to today's discussion.